So welcome to Journal Club. My name is Anahit. Next slide. So the article I'm presenting today is a preprint, and it's from a group from the McGovern Medical School in Texas, as well as the University of Texas Health Center at Houston. It was released earlier this year in April, and it's called Mitochondrial Health Index Correlates with Plasma Circulating Cell-Free Mitochondrial DNA in Bipolar Disorder. Next slide, please. So bipolar disorder is a severe chronic psychiatric disorder, and it's actually increasing in prevalence. It affects about 1 to 4% of the world population. And unfortunately, there's no sufficient treatment option as patients suffer symptoms or breakthrough episodes resistant to treatment. Now, mitochondria and BD have a clear connection. There's the mitochondrial dysfunction hypothesis that's been corroborated by several studies showing that BD patients present an atypical mitochondrial metabolism, oxidative stress, abnormal mitochondrial morphology and dynamics, and mtDNA damage. There's also been genetic studies that support this hypothesis, showing an increased likelihood of maternal inheritance and abnormal findings of the mtDNA of patients with BD. Finally, from environmental stress, there's chronic stress that can lead to an accumulation of mitochondrial dysfunction, which might represent an early event that increases allostatic load and disease risk in BD. Next slide. So just a background on MHI, that's the Mitochondrial Health Index. And the authors previously showed that BD patients have an impairment in MQC, which is the mitochondrial quality control. But this is the first study to investigate MHI in patients with BD and the association of a decrease in MHI with alterations in the MQC system, ccf-mtDNA levels, and clinical outcomes. So again, MHI is an index. It's the Mitochondrial Health Index, and it's composed of various parameters that Spring is circling on the slide. It's based on the science, I believe, developed at the University of Alabama based on their bioenergetic health index. But this one that the authors are using is based on Picard et al., which is the article snippet on the left. And this is the one that they developed. It mathematically integrates biochemical enzymatic activities and mitochondrial DNA copy number into a single score. And this represents mitochondrial function capacity on a per mitochondrion basis. Next slide. So the goal of this study was to evaluate MHI and BD subjects and non-psychiatry controls, and to determine whether lower MHI is related to a higher ccf-mtDNA level and poor clinical outcomes. So the hypothesis is that BD patients will show lower MHI due to the changes in the MQC network, which is associated with higher ccf-mtDNA levels and, again, poor clinical outcomes. So the study design included 14 BD patients, 16 controls, which were matched to the BD patients. And the samples included peripheral blood mononuclear cells, which were used to measure the enzymatic activities of citrate, synthase, and complexes 1, 2, 4, and mtDNA copy number. ccf-mtDNA was evaluated by qPCR in plasma, and MQC proteins were evaluated by Western blotting. Next slide. So they controlled for demographics and didn't find any significant difference when comparing BD and control patients when it came to these factors, with the exclusion of the psychiatric tests, MADRS, YMRS, and so on, as well as illness duration. But we'll get to those later. But they did control for these demographics, and they didn't find a significant difference. Next slide. So using one-way ANOVA after controlling for sex, age, BMI, and smoking status, they showed that patients with BD present lower enzymatic activity of the citrate synthase and complexes 1, 2, and 4 when compared to the controls, as you can see in the plot on the left. The present study showed that, the plot on the left shows that patients with BD had a lower MHI than non-psychiatric controls after adjusting for the confounding variables. And again, you can see what MHI is characterized as in the right figure. Next slide. So taking a deeper look at these results, you can see with complex 1, there's a significance of 0.002 when it came to the controls versus BD. Next slide. Again, significance with complex 2 with 0.001. Next slide. Significance with complex 4 with 0.001. Next slide. And citrate synthase with 0.032. Next slide. The one that didn't have any significance, though, statistical significance, was the comparison to mtDNA copy number with the value of 0.174. So there's no significance there. Next slide. So they also compared MIH, ccf-mtDNA with MQC proteins and active caspase 3. So MQC proteins, again, are the mitochondrial quality control proteins. They include MFN2, which is a protein essential for mitochondrial fusion. OPA1, a protein involved in the maintenance of the respiratory chain and membrane potential and control of apoptosis. And FIS1, a protein involved in mitochondrial fragmentation. And as we know, Parkin, protein that plays a critical role in ubiquitination. So the group previously showed that BD patients present lower levels of MFN2, OPA1, and Parkin. So you can see the values there of those three. While FIS1 was higher, you can see FIS1 on the right. Next slide. And here they also show P62, a protein for the degradation of ubiquitination cargo through autophagy, LC3, a key component of autophagy and mitophagy, and active caspase 3, which is involved in the execution of apoptosis in cells. So P62, LC3, and active caspase 3 were higher when compared to non-psychiatric controls. And you can see the significance when comparing the two groups. So again, they were higher when compared to the controls. Next slide. So because the MQC system is essential to maintain mitochondrial health, they also evaluated the relationship between MQC-related proteins and MHI. And the results show that subjects with higher levels of FIS1 exhibited lower MHI with a p-value of 0.001. Subjects with higher levels of OPA1 had a p-value of 0.001, and LC3 had a p-value of 0.001, and they had higher MHI. So you can see that those values there. ccf-mtDNA under the right columns had higher levels of FIS1 with a value of 0.003 and lower levels of OPA1 with 0.017 and LC3 with a p-value of 0.008. So those are the right values. Additionally, patients with higher levels of active caspase-3 had lower MHI with a p-value of 0.005 and higher ccf-mtDNA levels of 0.001. Next slide. Since there's an imbalance between MQC proteins and apoptosis activation, this can lead to the release of mtDNA into circulation. So they evaluated ccf-mtDNA and whether or not MQC-related proteins, active caspase-3, and MHI could be associated with ccf-mtDNA. And they found that patients had higher levels of ccf-mtDNA, as you can see in the plot on the left, which was significant. And in order to investigate whether MHI predicted ccf-mtDNA, they performed a linear logistic regression model, including age, sex, BMI, and smoking status as covariates. And they showed a negative significant predictive relationship between MHI and ccf-mtDNA after controlling for those covariates with a p-value of 0.001. Next slide. Now, I found this really interesting. In order to investigate whether MHI and ccf-mtDNA levels predicted mood symptoms and functional impairment, they performed linear regression relating to MHI and ccf-DNA to measure categories of mood testing. So that includes MADRS and YMRS. So MADRS is the Montgomery-Åsberg Depression Scale, and it comprises of 10 different items to evaluate mood. And the YMRS is the Young Mania Rating Scale, and it's a scale used to determine manic symptoms. They also looked at functional status, and they determined that through GAF and FAST. If you could go to the next slide, please. So these were tests to determine functional status, and GAF is the Global Assessment of Functioning Scale, and it's a scale to use to rate how serious a mental illness can be. And then FAST is the Functional Assessment Screening Tool Scale, which identifies factors that may influence problem behaviors. So if you could please go back a slide. So they looked at those functional statuses, and they also looked at clinical status, like length of illness, and they controlled for age, sex, BMI, and self-reported smoking status. So just looking at the measures of mood, you can see that there is that MHI and ccf-mtDNA predicted the severity of the depressive symptoms, while there was no significance found with the YMRS score. And you can see with the MHI, there's a negative predictive relationship between the length of illness and MHI. And that's the very bottom there. Next slide, please. There was a positive predictive relationship between length of illness and ccf-mtDNA, which is on the left, the first set of data. And this was after controlling for those covariates. And this suggests that length of illness may be partly responsible for the lower MHI and higher ccf-mtDNA levels observed in BD patients. There was similar analysis, again, controlling for age, sex, BMI, and smoking status in the combined BD and control samples to explore the association of MHI and ccf-mtDNA and functional status. Again, that includes that GAF and FAST tests. And it shows that participants with lower MHI and higher ccf mtDNA levels had worse functional status, highlighting the altered mitochondrial functioning, which can significantly cause functional deficits. So you can see that there's an association there between the FAST and MHI and ccf-mtDNA. Next slide, please. These are just the selected models that were presented that highlight the previous two slides. So you can see that all summarized here. Next slide. So just to summarize, lower MHI presents alterations in MQC protein levels found in the study. There's an increase in FIS1, and that presents lower MHI. There's higher MSI, which presents higher levels of OP1 and LC3. Mitofusion may account for this, for the higher bioenergetic capacity, but in contrast, mitochondrial fission is associated with a decrease in bioenergetic efficiency of the cells. Maintaining healthy mitochondria is regulated by MQC, and inhibiting the mitophagy pathway could lead to pronounced accumulation of damaged mitochondria. And these studies show that during the apoptotic process, fragments of mtDNA are released to the extracellular space and may behave as damage-associated molecular patterns, or DAMPs, and this leads to chronic inflammation without microbial infection, which is called sterile inflammation, and that's through the activation of various pro-inflammatory signaling pathways. They also saw higher ccf-mtDNA associated with lower MHI, MQC impairment, and apoptosis activation. Patients with BD had higher ccf-mtDNA, and this is negatively correlated with OPA1 and LC3, and positively correlated with FIS1. Higher MHI and ccf-mtDNA was associated with a higher active caspase-3 level. So BD patients have an increase in mitochondrial fragmentation associated with a decrease in mitochondrial bioenergetic capacity, following by a lack of mitophagy, which could lead to the accumulation of damaged mitochondria, shifting the cell signaling to apoptosis activation. So consequently, fragments of mtDNA are released into circulation, which trigger inflammation, and it's a consistent feature of bipolar disorder. Next slide. Increased evidence shows that BD is a systemic disorder, and not only affects the brain, but peripheral systems as well, leading to systemic comorbidalities that may represent the results of cumulative wear and tear from long-term stress. And as a result, they can influence and further deteriorate brain function, contributing to accelerated aging and neuroprogression. So in this context, this was just an exploratory analysis that investigated the correlation between MHI and ccf-mtDNA with depressive and manic symptoms and functional status. But they found that depressive manic symptoms were associated with a lower mitochondrial content and bioenergetic capacity and lower levels of ccf-mtDNA. And they saw that there was correlation between worse functional status with lower GAF and higher FAST scores, and lower MHI and higher ccf-mtDNA levels. And it's notable that previously they demonstrated that brain volume was a predictor of FAST scores in patients with BD, and could identify specific brain areas related to functioning impairment. And the FAST score was also reported as sensitive to detecting functioning impairments and accurate in distinguishing early from late stage BD. So that's notable. Length of illness was also found to predict MHI and ccf-mtDNA in BD patients. They found that there was a 1 unit increase in length of illness, resulting in a lower MHI of 1.949 and higher ccf-mtDNA of 26.42, again, after controlling for factors. Next slide. And this is my last slide. So there were some limitations to the study, including the possibility of type 1 error. Since there's a small sample size, there could be a false positive. Also, the single analysis of peripheral blood is not the most appropriate, as directionality and causality cannot be established. The study did not account for factors that could impact mitochondrial health, like lifestyle, diet, exercise, childhood trauma, etc. The use of PBMC cell mixtures could have reduced the ability to detect mitochondrial alterations. And there's some, like a specific purified cell could have had greater sensitivity and specificity among particular cell populations. Most patients were taking one or more psychiatric medications, introducing the confounding effects of polypharmacy. And there's also an absence of information on the number of previous episodes and hospitalization. So all in all, this study is just exploratory, and it would require further replication and validation. Future research efforts could look at the mitochondrial function and related aspects of cellular bioenergetics to investigate using a larger sample size, prospective longitudinal study designs, repeating measures from patients with BD during all affective states, and statistical adjustments for a range of relevant demographical and lifestyle variables, like I mentioned before, as well as replication.